Diabetes is a disease in which the patient has trouble regulating his or her blood glucose level. Until the early 1900s, this was a deadly disease. Imbalanced glucose levels would induce coma and eventually death. Arguably, one of the greatest medical discoveries of the 20th century was the development of genetic engineering technology. Let's imagine ourselves in the shoes of scientists struggling to develop a cure for diabetic-induced death. For some diabetic patients, uncontrolled glucose levels is simply the result of a lack of a hormone called insulin. Insulin facilitates glucose transport into cells, thus lowering blood glucose levels. If that's true, we should be able to treat this form of diabetes by providing the diabetic patient with insulin. Okay, easier said than done. Where are we going to get all this insulin? Scientists eventually determined that insulin purified from animals like cattle or pigs was sufficient to treat the insulin deficiency in these diabetic patients. While this was a major medical breakthrough, the animal substitute wasn't without its side effects. Some patients suffered an allergic reaction. As we will see, genetic engineering would eventually solve this problem and pave the way for an entire new field of scientific and medical possibilities. Genetic engineering is the process by which scientists modify the genome of an organism. Creation of genetically modified organisms requires recombinant DNA. Recombinant DNA is a combination of DNA from different organisms or different locations in a given genome that would not normally be found in nature. In most cases, use of recombinant DNA means that you have added an extra gene to an organism to alter a trait or add a new trait. Some uses of genetic engineering include improving the nutritional quality of food, creating pest-resistant crops, and creating infection-resistant livestock. Organisms whose genome has been altered are commonly referred to as GMOs. GMO stands for genetically modified organisms. But how does this relate to our insulin problem? We're struggling with the fact that some diabetic patients are experiencing an allergic reaction in response to insulin isolated from cows and pigs. Now, the obvious solution is to give patients human insulin instead of animal insulin. Injecting patients with purified human protein should minimize the allergic reaction patients are experiencing. But purifying insulin from human tissue is impractical. That's why cattle or pig insulin is being used at this time. Let's see how genetic engineering opened the door to mass-producing insulin rather than purifying it from animal tissue, human or otherwise. The organism that is modified in a genetic engineering experiment is referred to as the host. Depending on the goal of the genetic engineering experiment, the host could range from a bacterial cell to a plant or animal cell, or even a human cell. Now, you might guess that scientists chose a human or animal cell to produce insulin. If so, you may be surprised to discover that scientists instead used bacterial cells to produce human insulin. There are many reasons why bacteria play an important role in many genetic engineering experiments. Consider the space required to house larger animals. Bacteria are small and relatively inexpensive to maintain compared to most alternatives. Consider the time required to raise larger animals. Bacteria reproduce much more rapidly. Finally, consider the end goal. It is easier to overexpress and isolate molecules from a bacterial cell than it is from specific cells in a multicellular host. The vehicle used to transfer genetic material into a host organism is called a vector. Scientists typically use plasmids, viruses, cosmids, or artificial chromosomes in genetic engineering experiments. Let's discuss how a few of these vectors work. If you recall the biology of a virus, you can probably imagine how it could be used in a genetic engineering experiment to shuttle DNA from one organism to another. The virus basically serves as a biological syringe to inject DNA into the host cell. Normally, this injected DNA would lead to a viral infection. A genetically engineered viral vector, however, is designed by scientists to inject DNA of interest. In this case, it would be the human insulin gene. However, the vector chosen for the original insulin experiment wasn't a virus. Instead, scientists used a bacterial plasmid. A bacterial plasmid is a small circle of DNA typically found in bacteria that is separate from the majority of bacterial DNA located in the nucleoid. Scientists use plasmids frequently as a vector for DNA they are studying in an experiment. We'll explore the features and function of bacterial plasmids in another lesson. Suffice it to say for now that a plasmid is a vector that allows scientists to genetically modify a bacterial cell. In this case, so we can produce human insulin. In summary, genetic engineering is the process by which scientists modify the genome of an organism. Recombinant DNA is a combination of DNA from different organisms or different locations in a given genome that would not normally be found in nature. GMO stands for genetically modified organism. A host is the organism that is modified in a genetic engineering experiment. The vector is the vehicle used to transfer genetic material into a host organism. A plasmid is a small circle of DNA typically found in bacteria that is separate from the majority of bacterial DNA located in the nucleoid. In our insulin example, scientists created recombinant DNA by adding the human insulin gene to a bacterial plasmid vector. By inserting this plasmid into a host bacterial cell, the resulting GMO bacterial cell was genetically engineered to produce human insulin. We'll explore the specifics of how this can be accomplished in subsequent lessons.
History of genetic engineering. If we had to find one time to have as the beginning of genetic engineering, it would be 1953 with the discovery of the structure of DNA by James Watson, Francis Crick, and Rosalind Franklin. With the discovery of DNA, we understood that traits could be passed on from generation to generation, and that every living creature, it's built from this blueprint within them, within their cells, called DNA. And now that we know that this blueprint exists, we can start modifying the DNA to change the final product, to change the organisms, to have them do different things. A great example would be how genetic engineering totally revolutionized the production of insulin. Before the discovery of DNA, insulin used to be produced by gathering up cow pancreases and smashing them together and getting the juice and purifying it a little bit and giving it to people with diabetes. And a lot of people had allergic reactions, some people died, it didn't work too well on these people, and there was always the problem of production. You know, how many cows can you squeeze the pancreatic juices out of to make insulin? But now that we know about DNA, we don't need cows. We can just take the human insulin, we can take the letters, the code for human insulin, put it into any creature of our choice, and have that creature make insulin for us. And an ideal creature would be something that is really controllable, that we can grow lots of, and then we could collect and purify the product from. And they chose E. coli, and we take human insulin now, and we just put that writing into the DNA of E. coli and have the E. coli produce the insulin for us, and now it's very safe and it's readily available. So in that sense, genetic engineering, has really taken off because we've only seen the beginning. We've realized that our bodies, that all living things, plants, animals, creatures, microbes, are essentially little factories, and that the DNA is just schematics that we can feed into these factories and have them create whatever we want, And as the days pass, we're getting better and better at learning what we can create. What do we write to have it create this? What do we write to have it create this? A great example right now is many scientists are creating fuel, with microbes. They just program into the algae, they program into the E. coli, they program into the bacteria to make oil, and they do. They even program it into trees. There's a project going on right now in Brazil where they're growing, if you get syrup from the tree, you don't get sugars, you get turpentine that you can burn and use as gasoline. So it really is a way for us to harness the power of nature to really do what we need to do.
Have you ever heard about genetic engineering? Yes, this term may sound familiar to you. Genetic engineering is done to obtain a combination of desired traits in a similarly new organism. In general, the characteristics are aimed to give economic benefit. Genetic engineering gives advantages to our life. Look at the 2 rice plants. In general, rice plants are prone to pests such as insects that may eat up their leaves, reducing harvest results. Through genetic engineering, a gene which has a pest-resistant trait encoded to it is inserted to the rice genes, hence making the plant more resistant to pests. Another example of the advantage of genetic engineering is in the cattle farm. The economical value of these dairy cattle can be raised by injecting a kind of hormone called the bovine growth hormone. This hormone influences the cattle's genes, making them grow bigger and produce more milk compared to those not given the hormone. Can you see the salmon? Generally, salmon reach its full size in 2 to 3 years. However, with the help of a growth hormone injection which is derived from Escherichia coli, the salmon grows faster. Administered salmon will reach the ideal harvest size in just a year.
Parkinson's disease affects millions of people worldwide. The main symptoms are caused by degeneration of brain cells that control movement. These cells originate in the substantia nigra and communicate with the putamen and caudate. As these cells degenerate, they lose their ability to produce dopamine and to communicate properly with other cells. Current medicines improve Parkinson's symptoms for several years, but become ineffective as the disease progresses. An experimental biopharmaceutical drug called SARA-120 has been safely tested in over 60 people with Parkinson's disease. SARA-120 is intended to restore damaged dopamine cells and protect them from further degeneration. SARA-120 consists of a bioengineered viral vector containing only the gene for human nurturin, a naturally occurring neurotrophic factor. This viral vector serves as a carrier for delivering nurturin to brain cells. Neurosurgeons accurately deliver SARA-120 to the areas of the brain where neurons are degenerating. SARA-120 then enters the neurons, inducing them to produce a continuous, lifelong supply of nurturin. Nurturin is expected to restore the function of the neurons and protect them from further degeneration, hence potentially reducing the severity of symptoms and slowing disease progression. A new study is now underway to test the potential benefits and safety of SARA-120 in Parkinson's disease. For more information, please visit www.saragene.com.
Well, British lawmakers have approved a new technique meant to keep genetic diseases from being passed from mother to child. And it sounds good, but opponents say the controversial method raises many ethical concerns and could one day lead to designer babies. Caitlin Burke has more.
It's a technique that allows babies to be created from the DNA of 3 people, 2 women and 1 man. This sets the stage for the UK to become the first country with a law that allows embryos to be modified this way.
This isn't genetic modification. We're not altering the nucleus. We're not altering those 50% of genes that come from each parent. It's 0.054% of the genome, and there are only 37 genes to give energy to the cell.
The technique requires an embryo to be altered before it's transferred into the mother, a move currently forbidden by British law. One House of British Parliament just approved the measure, and if the House of Lords also votes to allow it, the first baby with 3 biological parents could be born as early as next year. But candidates for these specially created babies must have a specific problem: faulty mitochondria.
When you have a baby and find out that your baby's ill or got something that you've got, and life expectancy for a child in mitochondria is very short, very short. And I think that's devastating for a woman.
Mitochondria are structures outside the cell's nucleus that help produce energy for the cell. Mitochondrial defects can result in diseases like muscular dystrophy, heart, kidney, and liver failure, and severe muscle weakness. But opponents say the DNA swapping technique isn't safe or ethical. The group Human Genetics Alert says the move would open the door to further genetically modified children.
This would be the first time that we would intentionally manipulate the human genome. It's something that governments around the world have agreed for the last 20 years that we shouldn't do, and once you cross that line, then it's very hard to stop going down the slippery slope to designer babies.
The scientists behind the new technique say it would likely only be used in about a dozen British women every year. Caitlin Burke, CBN News.
We'll have more on this brave new world of creating babies in just a few minutes, taking a closer look at the new trend of young women freezing their eggs.
Our mission is to build a detailed, realistic computer model of the human brain. And we've done in the past 4 years a proof of concept on a small part of the rodent brains. And with this proof of concept, we are now scaling the project up to reach the human brain. Why are we doing this? There's 3 important reasons. The first is it's essential for us to understand the human brain if we do want to get along in society, and I think that it is a key step in evolution. The second reason is we cannot keep doing animal experimentation forever. And we have to embody all our data and all our knowledge into a working model. It's like a Noah's Ark, it's like an archive. And the third reason is that there are 2 billion people on the planet that are affected by mental disorder. And the drugs that are used today are largely empirical. And I think that we can come up with very concrete solutions how to treat disorders. When you walk up to a door and you open it, what you compulsively have to do to perceive is to make decisions. Thousands of decisions about the size of the room, the walls, the height, the objects in this room. 99% of what you see is not what comes in through the eyes. It is what you infer about that room. Can the brain build such a perception? Is it capable of doing it? Does it have the substance to do it? And that's what I'm going to describe to you today. So it took the universe 11 billion years to build a brain. It had to improve it a little bit. It had to add to the frontal part so that you would have instincts because they had to cope on land. But the real big step was the neocortex. It's a new brain. You needed it The mammals needed it because they had to cope with parenthood, social interactions, complex cognitive functions. So you can think of the neocortex actually as the ultimate solution today of the universe as we know it. It's the pinnacle, it's the final product that the universe has produced. It was so successful in evolution that from mouse to man it expanded about 1,000-fold in terms of the numbers of neurons to produce this almost frightening organ structure. And it has not stopped its evolutionary path. In fact, the human neocortex and the human brain is evolving at an enormous speed. If you zoom in onto the surface of the neocortex, you discover that it's made up of little modules, G5 processors like in a computer, but there's about 1 million of them. They were so successful in evolution that what we did was to duplicate them over and over and add more and more of them to the brain until we ran out of space in the skull and the brain started to fold in on itself. And that's why the neocortex is so highly convoluted. We were just packing in columns so that we'd have more neocortical columns to perform more complex functions. So what we did was, for the past 15 years, was to dissect out the neocortex systematically. It's a bit like going and cataloging a piece of the rainforest. How many trees does it have? What shapes are the trees? How many of each type of tree do you have? Where are they positioned? But it's a bit more than cataloging because you actually have to describe and discover all the rules of communication, the rules of connectivity, because the neurons don't just like to connect with any neuron. They choose very carefully who they connect with. It's also more than cataloging because you actually have to build three-dimensional digital models of them. And we did that for tens of thousands of neurons, built digital models of all the different types of neurons we came across. And once you have that, you can actually begin to build the neocortical column. And here we're calling them up. But as you do this, what you see is that the branches intersect actually in millions of locations. And at each of these intersections, they can form a synapse. And a synapse is a chemical location where they can communicate with each other. And these synapses together form the network, or the circuit of the brain. So let's switch it on. But to do it, what you have to do is you have to make this come alive. We make it come alive with equations, a lot of mathematics. And in fact, the equations that make neurons into electrical generators were discovered by 2 Cambridge Nobel laureates. So we have the mathematics to make neurons come alive. We also have the mathematics to describe how neurons collect information and how they create a little lightning bolt to communicate with each other. And when they get to the synapse, what they do is they effectively, literally shock the synapse. It's like electrical shock that releases the chemicals from these synapses. And we've got the mathematics to describe this process. So we can describe the communication between the neurons. So there literally are only a handful of equations that you need to simulate the activity of the neocortex. But what you do need is a very big computer. And in fact, you need 1 laptop to do all the calculations just for 1 neuron. So you need 10,000 laptops. So where do? You go to IBM. And you get a supercomputer because they know how to take 10,000 laptops and put it into the size of a refrigerator. So now we have this BlueGene supercomputer. We can load up all the neurons, each one onto its processor, and fire it up and see what happens. Take the magic carpet for a ride. Here we activate it, and this gives the first glimpse of what is happening in your brain when there's a stimulation. It's the first view. Now, when you look at that the first time, you think, my God, how is reality coming out of that? But in fact, you can start, even though we haven't trained this neocortical column to create a specific reality, but we can ask, where is the rose? We can ask, where is it inside? If we stimulate it with a picture, where is it inside the neocortex? Ultimately, it's got to be there if we stimulated it with it. So the way that we can look at that is to ignore the neurons, ignore the synapses, and look just at the raw electrical activity, because that's what it's creating. It's creating electrical patterns. So When we did this, we indeed, for the first time, saw these ghostly-like structures, electrical objects appearing within the neocortical column. And it's these electrical objects that are holding all the information about whatever stimulated it. And then when we zoomed into this, it's like a veritable universe. So the next step is just to take these brain coordinates and to project them into perceptual space. And if you do that, you would be able to step inside the reality that is created by this machine, by this piece of the brain. So, in summary, I think that the universe may have, it's possible, has evolved the brain to see itself, which may be a first step.
Cloning 101. The ability to create a clone used to be science fiction. Dr. Ian Wilmot's group changed that in 1997 with the creation of Dolly the sheep. Since then, the debate on applying the technique to clone human beings has been ongoing. Based on all the discussion, it may sound like cloning, human or otherwise, is an easy process. Find out what is actually involved in the cloning process by viewing our short animation. What are clones? Simply put, clones are organisms that have identical genetic material. In other words, the sequence of bases in their DNA is exactly the same. Long before the birth of Dolly the sheep, clones had been observed in both nature and in the laboratory. When a couple has identical twins, or triplets for that matter, the children are clones of one another. A plant cutting can also be used to generate a clone. Prior to 1996, it was thought that cloning an entire animal could only be done with embryonic cells, cells present in the early stages of an organism's development. In the 1950s, scientists generated entire frogs from embryonic frog cells. After a small number of cell divisions, embryonic cells start to change into different types of cells that an organism needs, including cells that form muscle, blood, liver, etc. This process is called differentiation. Although each of these cells has the same genetic material, each cell can only access the genes needed for its particular function. Before the experiment at the Roslin Institute, it was thought that once cells differentiated, they could not be used to generate an entire organism. For instance, in a sheep, udder cells could generate other udder cells, but not an entire sheep. The scientists at the Roslin Institute solved this problem by growing sheep udder cells under starvation conditions. This put the cells in a state similar to embryonic cells. This is called the G0 state. An egg cell was taken from another sheep. The nucleus, which contains the genetic material, was removed from the egg cell using a very fine needle. They then used electric shock to fuse one starved udder cell with one nucleus-free egg cell. They made 277 of these fused cells. Although the egg cell came from a black-faced sheep, notice that the nucleus with the genetic material came from the white-faced sheep. These fused egg cells were then inserted into several different sheep. These surrogate mothers were also black-faced. Of the 277 fused cells, only one progressed to form a developed lamb. Dolly was born on July 5, 1996. Scientists found that Dolly had the same DNA as the udder cells she came from. She is a clone of these udder cells. Dolly has given birth to a lamb named Bonnie, produced the natural way. Other lambs have been born at the Roslin Institute through their cloning process. Some carry genes that will produce usable human drugs. A laboratory in Hawaii, run by Dr. Ryuzo Yanagamachi, was the second group to successfully clone an animal from an adult cell. They cloned mice using cumulus cells, a cell type found in the ovaries. The cloning method used by the lab in Hawaii was different in two ways from the method used to clone Dolly. First, the cells used to clone the mice were not grown in culture, but instead were used immediately. Second, the nucleus was removed from the cumulus cell and then directly injected into the egg cell. This egg cell's nucleus had already been removed. The Yanagamachi lab used coat color to track genetic heritage. The cumulus cell comes from an agouti, brown, mouse, and the egg cell comes from a black mouse. The egg cell now had the same genetic information as the nucleus donor mouse. The egg cell was then activated and implanted into a white host mother. On October 3, 1997, the host mouse gave birth to Cumulina, named after the cumulus cells she was cloned from. Cumulina is the same color as the mouse that donated the nucleus. DNA fingerprinting confirmed that Cumulina had the same DNA as the nucleus donor. The scientists have taken cells from Cumulina to make more clones. They have successfully made several generations of clones, and all mice seem normal. Dolly the sheep died at the age of 6. Since the world said hello to Dolly, several other animals have also been cloned. Both Dolly and Cumulina were cloned from cells in the female reproductive system. Cows have also been cloned using ovary and cumulus cells, with the same method that was used to clone Dolly. Pigs have been added to the cloned animal menagerie. Scientists hope to use cloned pigs to grow organs that can be transplanted into humans.
DNA stands for deoxyribonucleic acid. It is the genetic material found inside a cell. A person's DNA is unique to them, just like a fingerprint. The only people in the world who share identical DNA are identical twins. Small portions of the DNA are called alleles. When a scientist looks at a piece of DNA, they can analyse the alleles they find. The alleles are identified and given a unique number. These numbers represent a person's DNA profile. At each location on the DNA molecule, a person has 2 alleles. Therefore, when looking at a person's DNA profile, you will see 2 numbers for each part or location of the DNA analysed. At the point of conception, a child's DNA is formed. Half of the DNA comes from the mother, in the egg, and half comes from the father, in the sperm. Therefore, when we look at a child's DNA profile, one of the numbers at each of the locations will match one of the numbers at the same location for each biological parent. Half of the alleles, or numbers, will match exactly to the DNA of the biological mother, and the other half will match exactly to the biological father. When testing other relationships, such as potential siblings, test participants are not expected to match at all locations. These tests are based on the fact that biologically related individuals will have more DNA in common than those that are unrelated. The amount of common DNA depends on how closely the individuals are indeed related.
Hello friends, welcome back to Career Fundas. Today we're going to talk about genetic engineering. Genetic engineering is a set of laboratory techniques for isolating genetic material from organisms. cutting and rejoining it to make new combinations, multiplying copies of the recombined genetic material, also called recombinant DNA, and transferring it into organisms, bypassing the process of reproduction. Genes can be exchanged between species that would never inbreed in nature. Thus, spider genes end up in the goat, human genes in plants, mice in bacteria, and bacterial genes in plants. Genetic engineering is the most powerful tool for biotechnology. Genetic engineers almost always work in laboratories. Their work demands great attention to detail and a strict adherence to safety standards at all times. Application of genetic engineering could repair a defect, as with the early trials of gene therapy in humans, enhance an effect already natural to that organism, such as to increase its growth rate, increase resistance to damage, increase resistance to disease or external damage, as in crops due to cold or drought, or it could enable it to do something it would not normally do. For example, getting a microorganism to produce human insulin for diabetics, or a sheep to produce a human blood clotting protein in their milk, in both cases a transgenic method, or getting a tomato to ripen without going squishy. Patience, an analytical and investigative mind, good laboratory skills, strong problem-solving skills, excellent oral and written communication skills, the ability to liaise with a wide variety of technical colleagues, the ability to work effectively as a part of a team, and good IT skills are important for an aspiring genetic engineer. Qualification or training. With subjects like physics, chemistry, mathematics, and biology in +2, your further study options could be BSc or BS in biological or life sciences, agricultural or biotechnology, and allied science with the goal of taking up genetic engineering. Alternatively, aim for an integrated 5-year MSc or MS in the biological or life sciences or biotechnology, or a medical science degree, or BTech biotechnology. Genetic engineering may be available as a specialization within BTech biotech in some universities. Few universities would offer a genetic engineering degree program. Genetic engineering is the most powerful tool for biotechnology, so biotech engineering is a useful base to chart a career in genetic engineering. Post-graduation and PhD in genetics or genetic engineering, or in one of the above-mentioned disciplines, would be advisable for an enduring career as a genetic engineering research scientist. Prominent institutions. BTech or BE in biotech at any of the Indian Institutes of Technology or IIT. Anna University, Chennai, Delhi University, Manipal University, VIT University, Vellore. Employability. Genetic engineering is a young, exciting, and controversial branch of the biological sciences. It offers the possibility of cures for diseases and countless material improvements to daily life. However, the frightening part of genetic engineering is its potential for misuse, either in schemes for population control or bungling that might produce a biological holocaust caused by a man-made virus. Postgraduates and doctorates in agriculture, medicine, biological sciences, bioinformatics, and allied disciplines may be working as genetic engineers. Genetic engineers work in research institutions, biotechnology, food processing, pharmaceutical, agricultural, or allied industries or companies, in universities, and in laboratories. Technical writing, scientific journalism, patent examining, and publishing could be other avenues of employment. Advances in biotechnology and medicine are driving a greater demand for individuals in this specialty.